right now. Now, microorganisms, basically, if you were to look at the microorganisms, they can only be seen with the help of a microscope. Some of the microorganisms are harmful to us. Yeah. The microorganisms like certain kind of bacteria and certain kind of fungus can make our food go bad. It leads to food poisoning. Some of the microorganisms can cause diseases in humans. Not just in humans, it can cause disease in plants and some other animals as well. Getting it? For example, common cold. You all must have suffered from common cold, right now. At some time, you must have suffered from common cold. Then you have we have diseases like malaria or skin infections. How many of you have heard of typhoid? Typhoid, you all have heard of typhoid?
Yes, sir.
Tuberculosis, yeah, or cholera,
Sir.
chicken pox, smallpox, AIDS. All of these are diseases that are caused by the microorganisms. Getting it now? So microorganisms, some of the microorganisms are harmful to us. Well, some of the microorganisms are also useful to us. For example, microorganisms like some bacteria, it helps in making curd and cheese. Getting it now? You all know what curd is. To make curd and cheese, you need some bacteria, or in order to make bread or cakes or pastries or alcohol or vinegar or some medicines also, we take help of some microorganisms, right now. Apart from that, there are some microorganisms which helps to decompose the organic waste of dead plants and animals. So you see that if there's a dead plant or dead animal in the surrounding or in the garden or some anywhere, you will find that after a week or so, you won't find it anymore there, because certain decomposing bacteria will start to eat them, right now. So dead plants and animals, they become food for the bacteria. So bacteria are helping in cleaning the environment also. What they do, they basically break up the organic waste into simple substances. So basically, they are feeding upon the organic waste. They feed upon the organic waste, and thereby they help in cleaning the environment. Now, you know, you guys must have heard of nitrogen cycle. How many of you heard of nitrogen cycle? Yes, Sundus, Khadija, Gauri. How many of you have heard of nitrogen cycle? None of you.
No,
So
sir. But I don't know what does happening.
see, basically in nitrogen cycle, what happens? I think by as of now, you guys must be aware of some common things. Like a plant requires certain kind of elements, like it requires nitrogen, it requires phosphorus, it requires potassium. Apart from these three, there are so many other nutrients required by the plants. One of them is nitrogen. So where does the nitrogen, how the plant takes up nitrogen? So see, basically, either we provide manures or fertilizers, we add manures and fertilizers to the soil, and the roots will be absorbing those minerals. It will be absorbing the nitrogen from the fertilizer and manure. But in some cases, we have plants like peas. If you were to look at the pea plant, and you look at its roots, so you will find that its roots have got a bulb like structure on them, which you call as root nodules. This is called as root nodules, and inside those nodules there's a bacteria found inside that nodule called as rhizobium bacteria. Anyone who has heard of rhizobium bacteria?
So,
Yes,
yes,
sir.
so, so you are
uh,
talking about this one, like this
which
whole
one?
stuff.
Yes.
So you're talking about this all stuff, like NPK and all.
Yeah, exactly.
So we know that.
Exactly. You guys have studied that, no? Like in the first chapter I did told, in the case of crop rotation we studied that. So basically this bacteria, rhizobium bacteria, is helping to fix the atmospheric nitrogen into the root. Huh? So bacteria helps in nitrogen cycle. Certain kind of bacteria helps in nitrogen cycle. Gauri, are you understanding what we are discussing?
Yes, sir.
So not only bacteria helps us in manufacture of alcohol, manufacture of vinegar, cakes, pastries, antibiotics, but also it helps in the case of nitrogen cycle. Right now, also it helps in recycling carbon. It helps in recycling of carbon also. Like you know that we humans are also made up of different kind of compounds. Like our body also consists of carbon in it. Right now, that's how carbon printing is done. You guys understand? uh, how we find the age of something? You guys know how do you find the age of how old something is? So radiocarbon dating is done. You guys have heard of carbon dating or not?
No, sir.
See, carbon dating is basically the process in which using which you are able to find the age of some living organisms. Suppose today I were to find the skeleton of some living organism. Suppose I find the skeleton. So how can I tell how old this skeleton is? What method do I have to find how to find the age of that skeleton? I would do the process of carbon dating, in which I would be studying about the carbon found in that skeleton. So what we can basically say that our body consists of compounds of carbon. So
Good,
scientists
good evening,
study.
sir.
Yeah, Chakrika, good evening. How are you?
I'm fine, sir. How are you?
Yes, I'm also good. So Chakrika, we are currently doing the chapter microorganisms, friends and foe. Okay now.
Okay, sir.
Do let me know what's going on in your school, by the way.
So we recently completed coal and petroleum,
Hmm.
and today we started cell.
Cell is going on. Okay.
Yes, cell structure.
What about the first chapter, crop production and management?
No, sir, ma'am didn't take that.
Okay, okay. So your school is also not following a proper sequence of the chapter.
No, sir.
Okay, okay. So see, what basically, as I told to Gauri also, so for you, I'm repeating for you, Gauri, and for you, Chakrika, that right now in the batch, this chapter is going on. Getting it now. Now,
Yes,
since
sir.
uh, in your school, chapter cell is going on, or is it completed? It's going on, no? The chapter cell structure and its function. That chapter is going on in your school, Chakrika.
So the thing is, today we had observation in our class.
Hmm.
So ma'am started the chapter, but she didn't really teach anything. She just made us do activities. That's it.
Okay, okay, okay. So see, Gauri, you also are having this chapter going on in your school now, I think.
Yes, sir.
Okay, so what we will be doing now? We would be, I would be scheduling class for you, Chakrika, and for you, Gauri. Okay, na? So there you guys would be studying that chapter from me. Okay. While in the
Yes,
Yes, sir.
batch, this chapter will be going on right now. So in a week, there will be one class in which I will be teaching you the chapter which are going on in your schools. Getting it now? Okay.
okay, sir.
Any questions?
No, sir.
So basically, what we were discussing
Sir, I
here
have a
now.
doubt.
Yes.
So basically, you'll only take the chapter which we have for which are going in our school, like every week.
See, basically, as I said, like normally there are 3 classes in a week. Right now,
Yes,
there
sir.
will be 3 science classes in a week. So you will be attending those 3 classes in the batch, right? Apart from these three
Okay, sir.
classes, I would be scheduling one class right now where I would be teaching what's going on in your school, so that you don't have a problem, like so that you don't miss out in your
Yes,
school.
sir. But the thing is, I'm asking if you'll only be teaching it for this week, like you'll only be taking the class only this week, or will you
No, no,
also
no.
be continuing it the other week?
I will be continuing it until the chapter is finished. Okay, no?
Okay, sir. Okay, sir.
Mhm. Yeah, so basically we were talking about microorganisms, and we were talking about what are the benefits and what are the harms by the microorganisms. So we were talking like some of the microorganisms can cause diseases like this, typhoid, tuberculosis, or cholera, or AIDS, while some of the microorganisms are helpful to us, like they help in the preparation of curd, in the preparation of alcohol, cheese, antibiotics. Some microorganisms helps to keep the environment clean also. They help in cleaning the environment by decomposing the organic waste into simpler substances. How they do so? They are basically feeding upon it, and in the process of doing so, they release certain chemicals which converts the organic waste into simple substances. Then we did a study that, like you guys already know about rhizobium bacteria that is found in the roots of leguminous plants like a pea. So rhizobium bacteria was known to take up the atmospheric nitrogen. It takes the nitrogen from the atmosphere and converts it into compounds like nitrates and nitrites. It converts it into NO2 and NO3. And this is not taken up by the plant. Apart from that, some microorganisms helps in recycling the carbon also. How they do so? Like, organic waste like plants or animals, they also consist of compounds of carbon in them, isn't it? If I were to study the cells of my body, if I were to study the cells of my body, I will find that there are compounds of carbon found in my body also. So any living organism that you see on earth, it will be having compounds of carbon in their body. Getting it what I'm trying to say?
Yes, sir.
Then I did talked about carbon dating. What is carbon dating? In carbon dating, suppose you were to found skeleton of some living organisms. So let's say that was living a very long time ago. So how would you find how old is the skeleton of that living organism? You would do this process called as carbon dating, in which basically we study about the type of carbon found in them. How old that carbon is can be determined by a method called as carbon dating. So once you determine the age of carbon found in it, we'll be able to quote the uh, age of this skeleton. Hope you guys are getting it.
Yes, sir.
So general discussion,
Sir, I have a doubt.
basically, yeah, Chakrika.
So, but during like in mortuaries, basically in postmortems, they are able to tell the time, exact time when they have died. But I mean, um, they're still not decomposed. Like their flesh is not decomposed yet, right? Like when they're just okay. For suppose, let's say they just died and they've taken to the hospital, mortuary, everything done, but they've
Mhm.
also submitted the postpartum report, and it and there'll be and the time will be exactly mentioned, right?
Yes.
So by that time, the dead body's flesh will not, it basically it won't be decomposed, right? As it's not buried yet. So how do they find it that time?
So basically, how in postmortem they determine the time of death.
Yes, sir. When it's like when the flesh of the person has not been decomposed.
Okay, okay. See, basically, once the body starts to, once the body starts to, once the body cells start to stop functioning. See, if you were to look at the human body, suppose there's a human body and it got shot. It got shot by a bullet. So a wound will start to develop at that place right now. So certain things must be considered in order to determine the time when how in order to tell the exact time. You see that so many different kind of determination is very important in this case, although the exact time of death cannot be determined for very sure. They whenever they say that, they will be giving a rough idea. It basically gives you a rough data. It will not give you a very accurate data. It's not like 11:33 p.m. It can't give you an exact data. It can give you a data between 11 to 12 p.m. like that. Getting it?
Yes, sir. But
So
even that
several
time, like, how are they able to find it
yes,
out?
how they are able to find out, there's different factors that they will be considering. They would be considering the temperature of the body, like what is the temperature of the body when, like when the body is handed to the postmortem department. At that time, they would be measuring the temperature of the body. One thing. Another thing, they would look at how much of tissues have decomposed right now. How much of tissues have decomposed. That is another thing that they will be considering right now. So basically, what
Yes, sir.
happens, the temperature of the body starts to fall. Another thing is that they would be considering about the fat also. Like gradually what happens, fat also starts to decompose. So they would be taking samples from different parts of the body. They would start to analyze it, look at the temperature. So different things they consider while determining it. Okay, no?
Okay.
Hmm. So hope you guys are getting it. We did talked about this scientist in the previous class. His name was Anton von Leeuwenhoek, and he's basically known as the father of microbiology. Reason being that he discovered the protistas and the bacteria. What this protista is? See, basically, I have previously also told this thing. Let me repeat that basically we have categorized the living organisms into 5 kingdoms. You have plant kingdom, you have animal kingdom, you have kingdom Monera, then you have protista, and you have fungi. These are the 5 kingdoms of living organisms that we have on Earth. So any living organisms, you see, it will belong to any one of the group here. So the person Anton von Leeuwenhoek, he has been called as the father of microbiology. Basically, you should understand the term microbiology. Basically, that branch of biology where we study about microorganisms. So he had done some very credible work in the field of microbiology, and he discovered the protista and the bacteria. So hope you're getting where this protista word came from. I did told about this also. Aristotle has been called as the father of biology, but talking about the father of modern biology, Charles Darwin has been called as the father of modern biology. Another important thing, microorganisms have been categorized mainly into 4 groups. Right now, they have been categorized into 4 groups, or some books would be mentioning them into 5 groups right now. So nevertheless, we would consider the 5 group classification of microorganisms. The 5 group classification of microorganisms are as follows. Anyone who knows what are the 5 group classification? The first one is bacteria. Second one is viruses. Third group is protozoa. Protozoa or protozoans. The fourth one you have fungi. And the 5th group of microorganisms we have got algae. So whatever kind of microorganism we are going to study, they would be fitting in either of these groups. Another thing you need to know that microorganisms may be unicellular, that is having just one cell, or they can be multicellular also, right? Now, also you might see terms like bacterium also sometimes. So what is the difference between bacteria and bacterium? Bacterium is basically the singular of bacteria. Okay, now just like viruses, so its singular is virus, as simple as that. Or when I say protozoan, protozoan is basically the singular form of protozoa. Okay, now fungi, the singular form of fungi is fungus. Okay, now and then the singular form of algae is alga. So hope these basic things are clear to all of you. Okay,
Yes,
let's
sir.
understand about bacteria. Talking about bacteria, bacteria are very small single cell microorganisms, and they have cell walls, but they do not have a organized nucleus and other structures. I repeat, bacteria are very small single cell microorganisms, right? Just like a plant cell has got a cell wall, bacteria also have a cell wall, while they will not be having an organized nucleus. See, if you were to look at the organization of the cells now, there are basically two types of cells. One is the prokaryote, another one is called as eukaryotes, right? See, if you were to look at the cells of this, look at this region over here, this red one where I'm pointing. This is the nucleus of this bacteria. This is the nucleus here, but it is very unorganized. It is not organized. It does not have a boundary. Getting it now? So it does not have a clear-cut boundary. So it kind of looks like this. It is dispersed. While if you were to look at cells of other organisms, you will find that their nucleus is very well defined like this. So out of the two, which one is well defined? Obviously the second one, right? Now, so what you find here, that we see that bacteria are unicellular prokaryotic microorganisms. So prokaryotic are basically those microorganisms which do not have a well-defined nucleus found inside them. Are you guys getting it?
Yes, sir.
Prokaryotes and eukaryotes,
Eukaryotic.
right now. So you see that basically those kind of living organisms that developed almost 2 to 3 billion years ago, their body structure was very simple. Scientists do believe that prokaryotes were the first form of life to begin on Earth. Understand what I'm trying to say? I'm saying that scientists do believe that prokaryotes were the first form of life to have occurred on Earth almost 2.7 or 2.8 billion years ago. So initially we had prokaryotes, the examples of which are as follows, like bacteria, which still is a prokaryote. Then you have archaea. These are the examples of prokaryotes. Now, over the time period of almost 2.7 billions, what we saw that the different kind of prokaryotes, they started to develop. So they developed into eukaryotes. So what is the simple difference between the prokaryotes and eukaryotes? Let's understand this. If you were to look at the cell of the prokaryotes, you look at the cell of the prokaryotes, the cells would lack a proper nucleus. The nucleus would be kind of like this in them. This is nucleus. Now you look at the cell of the eukaryotic organism. So you will find that it has a very well defined structure. The nucleus is having a boundary. So nucleus is surrounded by a boundary. While can you find any boundary in the case of a prokaryotic cell? No,
No, sir.
it is very unorganized, right? No. So the major difference between the prokaryotes and eukaryotes is that they do not have a boundary. The nucleus does not have a boundary, right now. While in the case of eukaryotes, they have a boundary. The nucleus have got a boundary. And prokaryotes, the examples of these are bacteria and archaea, right now. Now, if you were to look at the cells of your body, you take sample of any cell from the human body, cells of bones or muscles, any cell, you will find that all the cells will have a very well defined nucleus. It will have a very well defined structure. But those of prokaryotes, they do not have a well organized structure, right now. So the primary difference between the prokaryotic and eukaryotic cells is that they is that of nucleus, right? Now, inside the cell, you guys know that it's not only nucleus that is found inside the cell. There are some other components also found inside the cell. Like just in the human body, what do you find? There are different kind of organs. Different organs are there, like heart is there, kidney is there, lungs is there, stomach, intestines, etc. You have a brain. In the similar case, if you were to look at the cells, inside the cell, you will be having a nucleus that is called as the brain of the cell.
Sir, I
Inside
have
the, yes,
normally how much like a cell has a lifespan?
lifespan, it varies. It varies from organism to organism. It varies from organ to organ. Like bacteria comparatively have a very small lifespan. Bacteria have a small lifespan. While talking about the in the human cell, you see that skins also have a small lifespan. Like normally, you know that most of the dust that accumulates on the bed, it's actually the dead skin cells. Getting it? Most of the dirt that accumulates on the bed, how your bed becomes dirty, it is actually because of the dead skin cells that falls on the bed. Then you look at the bone cells. So bone cells have comparatively much more life as compared to it. So lifespan of cell varies from organ to organ. It also varies from one organism to another. Like bacteria have a less lifespan. It can have a span from 1 day to a month. Getting it? Okay, back to what we were discussing. Inside the cell, you will be having this nucleus that is referred to as brain of the cell. Then you have, just like we humans have got different organs that are performing different functions, like heart is pumping the blood, kidney is filtering the blood, etc., etc. In the similar manner, there will be certain kind of structures found inside the cell that are called as cell organelle. Yes, Gauri,
Yes,
Chakrika,
sir. Yes, sir.
they're called as cell organelle. So you
Yes,
have
sir.
some examples of cell organelle like mitochondria is there, chloroplast,
Endoplasmic reticulum.
endoplasmic reticulum, very good. Chloroplast found in the plant cells. You have vacuoles, right now, ribosomes, etc. So what you will find that in the case of the eukaryotic cells, those organelles will also be surrounded by a boundary. They will be surrounded by a boundary, right now. While you don't find that in the case of the prokaryotic cell. Is everyone understanding what I'm trying to say here?
Yes, sir.
Sundus, you getting it? Chakrika, Khadija, Gauri,
Yes, sir.
Yes, sir.
right now? So that is the difference between the prokaryotic and the eukaryotic cells. The nucleus is not organized. it does not have a body, neither does the cell organelles have body, while opposite is the case in the eukaryotic cells, right? So what type of cells we humans have?
Eukaryotic.
Eukaryotic. Okay, now? And the bacteria they have
Uh, Prokaryotic cell.
prokaryotic cells. Good. Right now, apart from that, you have archaea also. Archaea also. AEA actually. So hope that is clear. Now back to what we were discussing. We were talking about bacteria. So as I said that bacteria are unicellular prokaryotic organisms and they are very small. Another point about them is they are very small. Any questions so far?
No, sir.
Okay. Another thing I told you that they have cell walls. Okay, now. So plant cells and bacteria cells have one thing in common, that is
Cell wall.
cell wall. But the structure of the cell wall is different in both of them. Okay, na? Their cell walls are different from each other. Anyways, where the bacteria are found, so bacteria will be found almost everywhere in large numbers. It will be found in air, whether it be soil or in water. Every surface around us, every surface that you see around yourself, on that some type of bacteria will be present. On our body also some bacteria are present. Even inside our body, certain bacteria are present. Right now, can you guys guess a rough idea like how many bacteria are present inside our body? How many bacteria are present inside our body? Yes,
So many.
so many. Give a rough number.
Maybe millions.
Millions. It is actually trillion. Just imagine the number. See, if you were to look at the exact figure, there's around 39 trillion of bacteria found inside our body. Most of them are found in the intestine. Getting at what I'm trying to say, guys?
Yes,
Sundus.
sir.
Yes.
So you see, this many bacteria are found inside our body. So many of them are helpful, so many of them are bad. So many of them help in the process of digestion. Right now. So that is another figure. Note it down. Let me write it over here. There are around 39 trillion bacteria in human body. Let me erase other things which I have noted here. Now look at the four major types of bacteria. They are based on the shape and size. Bacillus, Vibrio, cocci, and Spirilla. Look at another slide. Here you can look at the different shapes of bacteria. Look at the comma shaped bacteria, spherical shaped bacteria, that is cocci. Then rod shaped bacteria, bacilli. These are the names of the bacteria that are having a rod shape. So they will be called as bacilli. One having a spiral shape will be called as spirilla. Okay, now? Getting it? Let me repeat again. You have the example of Bacillus, Vibrio, cocci, and Spirilla. So one that is having a comma shape, they are the Vibrio. Okay. Then you have cocci, Bacilli, and Spirilla. Examples of these shapes of bacteria are given as follows. Vibrio cholera. You have Vibrio cholera. You have Streptococcus, Salmonella, and Borrelia. Then look at the different kind of diseases that they spread. Vibrio cholera cause a disease called as cholera. Have you guys heard of cholera before? Yes. It causes cholera. Then Salmonella typhi, it is known to cause typhoid. As of now, just note down there, just keep in mind the shapes of the bacteria and their examples. So have a quick look at it. I will be asking few questions from it. Hmm. Give example of Vibrio, comma-shaped bacteria. Okay, you guys want to see at the slide once more?
Yes, sir.
Just look at it once more. Okay, these are the examples. Okay, give an example of Vibrio bacteria having a comma-shaped
Vibrio cholera.
Vibrio cholera. Good. Next, give me example of rod-shaped bacteria. Bacillus.
Salmonella.
Salmonella typhi. Bacillus-shaped bacteria. Salmonella typhi. Is it correct, Gauri? Yes.
I think no. Yes, sir.
What about spirilla and cocci shaped? Yes. See, for the bacillus, you had the example of E. coli. Getting it?
For spirilla, it's Borrelia.
Repeat again.
Borrelia.
For spirilla shaped bacteria, that is,
Borrelia.
I can't hear you properly. Repeat again.
Borrelia.
Chlor. What did you just say? Please repeat again.
For spirilla shaped bacteria, it's Borrelia, right?
See, basically, spiral shaped bacteria is okay. Borrelia, you are saying no? So Borrelia. See, it's Borrelia. Okay, no. Then talking about the cocci shaped bacteria. Yes, you have the example of. You had the example of this streptococcus. Okay, now. So what you guys will be doing after the class, you guys will be remembering the examples of these different shapes of bacteria. Okay. Now let's talk about the different uses of bacteria. What are some useful bacteria and what are some of the harmful bacteria that we have? So talking about uses of bacteria. Yes, you guys mentioned what are some uses of bacteria? Yes.
So they help in making alcohol.
In
Help in
bakery also.
baking, good. Baking cakes
Sir,
and
making curd.
in curd, very good. And
Medicine.
Sir, bread. Making bread.
baking cakes and bread, we can count them as one.
Okay, sir.
Any other uses?
Medicine.
Medicine, very good. Particularly in making antibiotics. Then, like
Sir,
it is, yes.
converting atmospheric nitrogen into dissolved nitrogen for plants.
Very good. That is in nitrogen cycle and carbon recycling. Then
Carbon dating.
um, that in itself it's not a help. Like we don't use bacteria to do carbon dating. Basically, in order to do carbon dating, you look at the concentration of different types of isotopes found in the skeleton or the remains of living organisms. That's a different things on this. Okay, now there you study about type of carbon found in the living organism and how old the carbon is. We don't take help of bacteria. Cleaning environment. Okay, now baking cakes, bread, curd. You can add in this cheese also. Getting it now. Then you have use of bacteria in agriculture also. Let me write it on the next page. We use it in the field of agriculture also.
So rhizobium bacteria, like that?
Rhizobium bacteria is there in nitrogen cycle, no?
So can it be counted?
Yeah, under this it can be counted. Like it will be helping in the nitrogen cycle.
Okay.
Okay, now. So you have example of biofertilizers. You guys must have studied about biofertilizer in the first chapter. Yes. Apart from this, nowadays we take help of bacteria in genetic engineering also. Okay, now in genetic engineering, these are some of the common uses of bacteria which we have, right? So bacteria. One more thing. Another important thing was about this sewage treatment plant. In sewage treatment plant also. Hmm. You guys know what a sewage treatment plant is? Basically, all your waste goes through a sewage treatment plant, right now. So all the domestic waste coming from the offices, industrial places, goes through the sewage treatment plant. So whatever biotic waste will be found in the waste, that will be decomposed with the help of certain bacteria. Okay, now. Now you'd be surprised to know a very interesting things that nowadays in the mining of certain kind of minerals, like mining of copper, mining of gold, or mining of other metals, there also we are taking help of some kind of bacteria. Ever thought about this? Like even in the mining industry also we'll be taking help of some bacteria. Might sound a little bit strange, but yes, we do use bacteria there also. So last point would be in recovery of gold, copper, and other metals in the mining industry. Okay. So you guys also, Chakrika, from now on, all of you are required to make notes. Okay, now. So I suppose in the previous class, you guys made few notes from this chapter. All of you are required to give the heading microorganisms, friends, and foe. If you guys have already given the heading, then ignore.
So
Yes.
actually, is it possible for you to send the PPT with everything written?
See, basically, from this class now, I'm basically thinking that it would be better if you guys make live notes of the class. Or if you guys have any suggestions, please do let me know.
No, so like I did attend the before classes, right? So
Yes.
I didn't attend before, right? Like the starting of this chapter.
See, in the last class, now, we did discuss the chapter only for like 10 or 12 minutes.
Okay,
So
so
it won't be much of a problem for you as well.
Okay, sir.
Okay. Or if you guys have any suggestions, please to let me know. Your suggestions are welcome. Sundas, Sundas and Khadija, particularly you guys, have you guys prepared notes for the first chapter, by the way?
Yes, sir.
Yes?
Yes.
Yes. Do one thing, give the heading in your copy and write this paragraph. Microorganisms, write whatever is written
I've
here.
already wrote, but I have wrote jumbled.
No, no, wrote it down once more.
Sir, I already wrote it.
You guys have wrote it down already. Okay, okay. Chakrika, can you take the screenshot? Gauri, you have also written it?
Yes, sir, just a second.
Okay. You guys will be required to just write this
Sir,
much.
actually, I can't take a screenshot for some reason.
Okay, okay. Anyways, okay, I will be providing the screenshots to you after the class. Okay, now.
Oh, okay, sir. Thank you, sir.
Meanwhile, you guys start writing from here. You guys just wait for 1 minute. I have repeatedly like there's an urgent call. Okay, now I will be back in a minute. Yeah, sorry for the interruption. I'm back. Okay. So, see, after the class, I will be providing the PDF of whatever we have discussed so far. Okay, now. So these are the different uses of bacteria. First of all, let's understand about alcohol production. How alcohol is produced with the help of the bacteria. How we take help of this. Getting it?
Yes, sir.
So you guys tell me, how do we use bacteria in the production of alcohol? Any idea? Like you have some kind of bacteria that basically will be helping you in the production. So what basically it does, like you guys must have heard about Pasteur, Louis Pasteur. So Louis Pasteur, he did took help of yeast and some bacteria to break down pyruvic acid and produce a alcohol. Like basically what he did now in the manufacturing of alcohol, you know how alcohol is manufactured. You would be taking different kind of fruits basically right now. So he basically took different kind of fruits. In that he added some amount of yeast and he added some amount of bacteria in that. So using that, he was able to break down the acid found in that to alcohol. So there it is used. But in the production of alcohol, yeast plays the main role. So apart from yeast, we just take help of some amount of bacteria. Okay. Then how do we use them in the baking industry? How do we use bacteria in the baking industry? So you have certain examples. Like you have certain examples of bacteria that will be taking help of, but particularly here also you will again be taking help of this yeast right now. Then there's a bacteria called as lactobacillus. Lactobacillus bacteria is used in making curd. Okay. Antibiotics. What are antibiotics? Basically, antibiotics are the weaker versions of the bacteria or some other microorganisms. Antibiotics are basically weaker version of the bacteria which have caused the disease right now. So there we use. Then like in the case of nitrogen cycle, we had the example of rhizobium bacteria. Then again, as we did a study in the carbon recycling, bacteria will be helping in decomposition and thereby basically will recycle the nitrogenous compounds found in the organic substances. Then helping in keeping the environment clean. So there also they will be helping. Talking about biofertilizers. You guys have heard of biofertilizers before? Yes. Like what are biofertilizers? Do you guys know about this?
Yes,
See,
maybe.
bio basically refers to anything that is living. Fertilizers, how can fertilizer be living? So here basically we are talking about particular some kind of microorganisms. So some of the bacteria are also acting as biofertilizers. Like you had the example of this rhizobium bacteria. You have the example of azotobacter. These are the bacteria that enriches the soil. Like we talked about rhizobium, it enriches the soil with nitrogen. Getting it? So these are basically called as biofertilizers, right now. So they help in promoting the growth of plant by improving the nutrient content of the soil. They enhance the productivity of the soil by fixing atmospheric nitrogen. Some of them will be helping in fixing sulfur into the soil. Getting it or not? So
Yes,
these are
sir.
some of the common biofertilizers which we have. Apart from azotobacter, you have other examples also. Like you have example of azospirillum. That is the case. Then talking about how bacteria are used in biotechnology. Right now, see how they are used in biotechnology or how they are used in genetic engineering. First of all, let's understand how they are used in biotechnology. So in the field of biotechnology, like they would be used to prepare antibiotics and other kind of medicines are prepared with the help of the bacteria. Now, not only bacteria, further when we talked about fungi, you would see that even some kind of yeast is also used, some kind of fungi is also used to prepare some medicines. Right now, let's understand about the use of bacteria in genetic engineering. You guys understand what is genetic engineering? Yes.
No, sir.
Genetic engineering. See, whenever some modification is done in the genes, like first of all, let me give you a background. See, this is a cell. Inside the cell, you have got nucleus. Inside the nucleus, you have got thread-like structures that are called as chromosomes. And in the chromosome, you have another substance called as genes. Getting at what I'm trying to say? Let me repeat. This is a cell. In the cell, you have a nucleus. Inside the nucleus, you will be having thread-like substances that are called as chromosomes. And in chromosome, you have gene. So what we basically do in the field of genetic engineering, we are making changes in the genes. Are you guys able to understand what I'm trying to say?
So
Yeah.
can you explain it again?
Yeah. Basically, I'm saying that what genes are, first you need to understand what genes are. Only then you will be understand what genetic engineering is. So genes are found inside the nucleus, and genes are responsible for transferring the character from the parents to the child. For example, if you were to look at a child, if the child is tall or whether the child is short, or whether the child will be having a curly hair or the color of the hair, all of this is determined from their parents right now. So if the parents are tall or short, they have curly hair or straight hair or whatever their color, skin color is. So these are the informations that are stored in the genes of the parents. So whatever character, whatever traits this child is receiving from his parents, from his or her parents, that that characteristics is stored in the genes found inside the cell. Are you guys able to understand what I'm trying to say?
Yes, sir.
Everyone in the class getting it?
Yes, sir.
So that is the thing. So if we are able to make some changes in the genes, thereby that is called as genetic engineering. So you have certain kind of bacteria like agrobacterium is there. So what basically happens in the case of the genetic engineering is that we would be modifying certain kind of bacteria. We would be making changes in their genes, so in order to produce large amount of proteins which can be used for different kind of purposes. So what happens, uh, different kind of bacteria will be grown in a very large quantity, and I'm talking about those bacteria whose genes have been modified, right? And you guys know that bacteria can be used in different purposes. Like in genetic engineering, what basically we'd be doing, we would be making changes in the genes of the bacteria, and once we have done so, we would be producing them on a large quantity. And once we do that, those bacteria can be harvested to get the desired type of substances from them. Like you can get desired kind of protein from them. Okay, na? See, basically this topic, this will require some time. So I think in just 1 or 2 minutes it won't be covered. I will be taking up this topic in the next class. Okay, na?
Okay,
So
sir.
that's it for today. If you guys have any other questions, please to let me know.
Yes.
Also, like genetically modified bacteria